Hello, welcome, greetings, salutations, and in the Gaelic, Ja Hoich. This is Communicating Your Data to Statisticians, a Biostatistics, Epidemiology, and Research Design Core Special Topics Talk number 4. I'm Dr. Mark Williamson for the BERD Core for DaCCoTA. All right, let's dive right in and get started here. So the goal of this talk is to improve communication between researchers and statisticians. So a quick note on my part, I am a statistician for the BERD Core, though my background is in biological research and even physics. So I come at statistics in a much more pragmatic way, that is, using it as a tool to get something done. And I find that this is often a helpful approach because a lot of researchers view this thing the same way. They don't really care about all the milieu of what's going on under the hood with statistics. They want to use it to answer important, interesting, fascinating research questions that they have. So I often start any sort of statistical approach by trying to answer sort of common sense questions. Like, for example, do I understand what the statistical method is doing? There might be a fancy, convoluted method to use, but if I don't really understand what it's doing, that's not very helpful. Another question is it interpretable? Can I understand what's going in, what's coming out, and what it all means? Thirdly, is it comparable to other studies? I don't like to work in a vacuum. I like to see what other people do to solve similar problems so that I know my approach that I'm using fits with what other people have done and had success in. And then finally, this is really the big key, is can I justify and defend it? Because no matter what you do at the end of the day with statistics, you want to be able to justify and defend your results, have reasons for why you did it, and why you think it is answering the question that you put to it. If you can do that, I think you've had a successful use of statistical analyses. So with all that in mind, I am not a master of all the theoretical sort of mathematical underpinnings of statistics. Statistics can be pretty difficult, but I do have a very good working knowledge of it and able to bridge the gap between using it to take data and turn it into results. So moving on here, part of the communication between researchers and statisticians is to be able to bridge the gap between experiment and analysis. Because a major obstacle is you have to move from the specific, the researcher side, where all the specific little details, unique setup of their experiment, their subjects, their data, whatever it is, to what I would say the more general on the statistician side. Well, what sort of data type is it? What sort of analysis it is? It's more abstract and then more generalized. So a target outcome of this talk then is to train researchers, to help give them tools to generalize their data so that statisticians can grasp it quickly and be able to approach the specifics of it. So a lot of how I've designed this talk is if I were to teach a researcher how to present their data to me, this is what I want them more or less to do. So hopefully that is helpful, not just for me communicating with researchers, it's for other statisticians, other researchers in general. And so the approach I'll be using today is what I call the 4 big topics. So let's take a look at what that means. So the 4 big topics are 1, big picture goal, 2, general test type, 3, variable information, and 4, sample data. Let's break those down a little bit before moving on and talking about them in more detail. So the big picture goal is the ability to be able to sum up your experiment first in a few sort of clear sentences. This would be like your explanation. A lot of times what goes into, say, an abstract. And then the second bit, which is the more important bit, is to be able to sum up your experiment in terms of X and Y variables. So we're summing up in the terms that a statistician we're actually using to build a model. So like X variable as a function of, Y variable as a function of X variables. And usually you don't need to specifically have it in a function like this, but you can have it in a sentence form. Number 2, general test type. This is what sort of test you're expecting to come out of it. And maybe you don't know this, that's fine. That's what, communicating with the statisticians can help. So the general style of test, really, when you break down, a lot of things end up being one of a couple of general types. Obviously, there's more complicated things, but it's always good to start there. And then also your discrete results. It's nice to be able to plot out or sort of imagine what your results might look like. 3, variable information. This is getting into that nice nitty-gritty, is to understand what variables you're using, like what data are you going to have to use. So name and type, so things like whether it's a number or a category, other sorts of measurement information. Do we know the general mean? Do we know the range? Like what does the data look like? And then 4, sample data, that sort of riffs off the third one, is if you have any preliminary or mock data that could really be used to help sort of build whatever analyses you want to do, be able to play around with that. Or if, barring actual data, if you have a similar experiment or know of a similar experiment you're working off of, even having that information helps statisticians to understand what maybe the approach would be or what the data might look like. So it might be something like that. So let's dive into more depth in each of these in the next slides here. So let's turn back our attention to the big picture goal. So as I said, the first part is to sum of your experiment in a few clear sentences. The idea of this, it helps translate the details into the big picture. Now you can give a statistician maybe your research proposal or whatever and whatnot, and they'll have all the details, which is fine, but it often can take time to distill out what's actually going to be done. And so something like more of like an abstract form can work well here. And keep in mind that maybe you don't just have one analysis you're going to do, maybe you have several sub-experiments to explain. So you want to be able to sort of detail all the little things you want to do. Not so much in every little detail, but every discrete analyses you're going to need done. Then the second part is some of your experiment in terms of X and Y variables. And I really like to be able to state this in like a sentence or two in the sort of form, some sort of similar form that I kind of have here. We want to model Y variable as a function of our X variables, or we want to see if X variables predicted the Y variable, or we wanted to see if the Y variable was affected by the X variable. Those all kind of mean the same thing or very close things, but you can kind of tweak it as it makes the most sense for your data. And of course, you can always add more information if you have it, like if you're correcting for certain variables or you're doing this across time, et cetera, on and on, so and so forth. So to get at a little bit the second part, which is a little more kind of abstract, I'll show a couple examples of actual research data I've done research on and how I might explain it in those terms. So I worked in this nematode research, and so I might say we wanted to model nematode growth rate, so that's our Y variable, as a function of gene copy number. Similarly, I looked at this nuclear reactor status. So we wanted to see if the presence of nuclear power reactors in a state, so that's our X variable, predicted brain cancer incidence, our Y variable. So you see that I use kind of this wording for here and this wording for here. And then finally, for COVID, we wanted to see if COVID-19 county death rates, so our Y variable, were affected by county rural urban stress. So again, I use it this way. So they're all, pretty clear, straightforward, gets the gist of what you're doing in one or two sentences. Now, obviously, these are pretty simplified. Like I only have one Y variable, one X variable. I don't show any of the other details. So obviously it might be a little more complicated, but this is a great place to start. Okay, so let's meander on to the general test type. So what I mean more specifically about this is, well, what type of test do you anticipate your experiments to be? And, again, maybe you don't know this, but it's pretty easy to sort of flesh out what it might be looking at your data a little more. So for example, if you're comparing your means across two groups, that's going to be something like a t-test. And so a great example of this would be like you have your treatment group and your control, and you have some sort of measurement. Maybe you're measuring blood serum levels, and you're going to have a couple bars looking between treatment and control. T-test. If you're comparing means across 3 or more groups, that's going to be ANOVA. So maybe you have a treatment, control, and maybe you have treatment 2, and maybe even treatment 3. I'm just going fast and loose as I go here, and so forth. And then on the other hand, if you're comparing 2 more numerical values, that's going to be something like a regression. Your classical scatter plot, you have your x variable, your y variable, and then a bunch of dots, and you're probably fitting a line. And if you're comparing frequencies across categories, so you have no, in a sense, numbers, you just have categories, that'd be a chi-square, something that's very common. You've probably seen in your genetics class, however long ago that was, is things like Punnett squares. You have frequencies of genotypes and phenotypes, and you might compare that, to another underlying pattern or so forth. And of course, these are just sort of like the most basic ones. There's obviously more complicated models, but so you might have a generalized or a mixed model, logistic, Poisson, or other regression. Maybe it's survival analysis. There's all sorts of much more complicated things, but a lot of these kind of get their start with a more simple setup. And then another thing here, you see here, I'm, as I'm going, I'm just sketching out these sort of expected results that kind of help visualize what the end goal might be. I find that that's very helpful for researchers to be able to do that. In fact, I've been working on some recent research where that's exactly what I did. I sketched out sort of what I expected my results to be, so I could have a goal to sort of aim at, a target as it was. So this will help you visualize the outcome and then how you need to get there. And a lot of times, this is probably above and beyond anything, most statisticians will ask for, and that's fine. None of these things for these topics are like, you must do every single one of these. These are sort of a collection of tools that are helpful, and some might not be applicable, or some might be above and beyond what the specific researcher needs. But they can be useful to think about. And these graphs can be as simple as hand-drawn graphics, as I've shown here, or they can be more complicated things here. So first of all, I'm going to just get out here because I have my examples I want to show you. Okay, so my first thing here is that nematode growth rate is a function of gene copy number. Well, I can do a regression because these are both numerical variables. My state nuclear reactor status, so that's with reactors or without. It's going to be this 2 groups, so that's a t-test. And then COVID across this rural urban county type, that's 9 different groups, so that's going to be more than a t-test, it's going to be ANOVA. And in fact, I sketched out, it helped that I've already actually done the research, so I knew what the data looks like on the other end, but these are just things I sketched out to kind of show what the results should be. Here's my regression, here's my t-test, and here is my ANOVA. And I, in fact, did all this actually just a couple minutes in a free software called GIMP. It's New Image Manipulation, either program or platform. So it's not too hard to be able to visualize your expected results pretty quickly. Next, let's turn our attention to variable information. So the first bit here is to write down the variable names and types. Now you might not know all your variable names and maybe not sure what's going to go into it, but you should have a good idea about the big picture stuff. So one thing to start out with is, first, it's very good to provide the full name rather than just an abbreviation. For example, this is data from the NHANES dataset. One of the variables is called LBXWBCSE, which is incredibly unhelpful. I have no idea what that means. But the description of it tells me exactly what it is: white blood cell count, which is much more helpful. And then anytime you can, if you include units, like what's actually being measured, that is also helpful. Because, for example, then, same example here, turns out that the count is 1,000 cells per microliter. It should be. So, after that, some more type. First of all, the two kind of big dichotomy, whether it's numerical or categorical, that is this number, or like it's a category, like some sort of group. And then from there, you can have more specifics. For example, numerical, is it continuous? That is, can it take in really any value? That's usually indicated by decimals in your data. Or is it discrete? So like counts, like 1, 2, 3, 4. It can only be sort of discrete numbers. And then with categorical, is it ordinal or nominal? This is kind of less important, but can still be useful. Ordinal is if you can order into useful categories like low, medium, high. And more nominal, there is real no order, like maybe color, like, red, purple, blue. I mean, unless you're doing electromagnetic spectrum, where therefore it corresponds to wavelength, usually color doesn't matter. But I mean, maybe a better example would be something like location or like state. That'd be a good example. And then, of course, in this first part, you can always include other considerations that you have that are more complex, like confounding variables or random effects. We'll touch base on those a little bit later too. And then the other bit here is writing down the variable measurement information. This is really helpful to kind of get a feel for what the data will look like. For the numerical, it's nice to have something along the lines of central tendency. This is like mean, median, mode, etc. Some sort of measure of spread, so like a standard deviation, variance, even range, even kind of min-max can be useful. And if you know it, it'd be great to know distribution, though that's not always known. For categorical, a little simpler, typically just like the number of groups and maybe the size of each group. And a big thing with here is to distill these variable information out. A lot of times what you'll really need to do is be able to translate from your method to your measurement. So a lot of times a method section will have things like, well, we're going to run a Western blot and stop it there. What this really translates to the statistician is that you're going to be generating these data that are protein concentrations, in like microliter. And now, as someone who's running Western blots, I know what the data will kind of get out from, but there might be other various and sundry measurements and experiments and all sorts of things to collect data that I don't know what the outcome will be. I don't know what they're actually measuring, so it's important to be able to translate that. So let's take a little look-see here, a little example by using these 3 sets of data that I've been talking about beforehand. So let's pop up a nice little blackboard. And so for the first one, so we have our first one is growth rate. There's several I did, but I'll be looking at the most interpretable one is the number of eggs per adult. So this is numerical and actually continuous because you could have decimal numbers. And the mean here I have is about 40 eggs per adult. But then the range is, I have the range here is some were like less than 1 all the way up to over 500. So huge, pretty range here. We can see that the mean is pretty low. So maybe even this seems kind of skewed towards the higher end. There's a few variables that skewed. So that's important to know when kind of figuring out how to analyze this. Now for my copy number, my gene copy number, is also numerical continuous, even though technically the data itself is discrete. You can't have like 1.5 copies of a gene. You can have either 1 or 2, and so forth. But since I'm estimating this, there are various things. There are actually decimals come in because like how you estimate this, it's not perfect. So I would consider it continuous. And it turns out the mean was about 166. And again, though the range was massive, and this is actually not true copy number, relative copy number, and again, there's a lot of error in it, but it ranged from 1 to 800. So it had the same sort of issue here where a few of the species of my nematode had really high numbers, while some had pretty low ones, and the means kind of show that it's skewed more towards, most have lower, but then there's few that like are overrepresented. So that's again important to know. Now let's move on to our state nuclear power reactor status. So our brain cancer incidence rates, that's again numerical and continuous. And here's some of the interesting things with measurement using the units. So this is like essentially mortality, so deaths per 10,000 people, and then these are age adjusted. I just read it with AA. And the mean here is 7.2. So that translates 7.2 deaths per 10,000 adjusted by age. And then the standard deviation, here's a measure of spread, is 0.37. So pretty tidy. And our reactor, so this is a categorical and ordinal, because technically you can order it like 0 and 1. Like there's yes and no. Like having ones is bigger than not having them. And there's just 2 groups, yes or no, and then so I'd call this binary. Okay. So now we have our final thing with covert cancer days. So first of all, I'll have my rural-urban status here. It's categorical and ordinal. Again, you can order it. There's going to be 9 groups from 1, the most urban, up to 9, the most rural. And then my mortality rates, again, it's numerical and continuous, sort of like our brain cancer rates, and it's again per 10,000, but it's not age adjusted. And then the mean's 12.92. And then here I had, instead of standard deviation confidence intervals, from 5.7 to 35.6. So here is example of how to kind of spit out some of our variable information. Okay, last big topic here is sample data. And this is definitely going to be the kind of the most, can be the most demanding portion of it, though not necessarily on your end, maybe more the statistician's end. But it can be really the most interesting stuff going on here and give us the best information of like how we're going to analyze this data. So first of all, if you can provide preliminary or mock data, that'd be the best. So preliminary data, maybe pilot data, sample data, whatever name you want to give it or it's given. It's great. It doesn't need to be a full data set, just enough to get a feel for analysis. So if there's something like a feasibility study with only a couple patients and say a clinical trial or something, if you have that data and you're setting up to do a bigger experiment, it's great to be able to look at that preliminary data and work with it. And of course, another important side note is having sample data is great for power analysis, to be able to figure out what maybe an effect will be of some sort of experiment, so you can accurately estimate how many samples you'll need to really detect a true effect. And of course, now you don't always have preliminary mock data. You can always try to create your own mock data through simulation. And that can be a pretty advanced topic. I have a nice little, just if you want to get your feet wet there here. But here I have an example. It can be as simple as something like this, where I make a y variable, which is normally 100 of them, normally distributed with a mean of 20 and standard deviation of 5. And then I'm going to round that to make it a little nicer. I can make an x variable that's going to be count, so from a Poisson distribution, don't worry too much about that, just that there's also 100 of them. And then I'm going to make a categorical variable, which is 4 different groups, A, B, C, and D, all of 25 per group. And then I'm going to put this in a data frame called sim data, and let's view it. just the first 12. You can see here that, oh, you simulate data in a couple lines here of our code. You can see that y variable, numerical continuous, x variable is numerical discrete, and then this is a categorical, and really it's nominal. Obviously, there's no real connection between y and x. If there's any real relationship, it's by random chance. You could, of course, do more complex simulation to link y and x to have some, like, x depend on y but to a certain amount, so on and so forth, depending on what your needs are. But this is a good, simple example to show that, yeah, you can in fact simulate data, and that can be very useful. Anyways, on to the second point here is another useful bit is to be sure a report of a similar experiment, whether you've done it yourself or you are using that as a basis for designing your own experiment. This can be very helpful. So like published papers that say you've referenced in your proposal, this is actually a great place to go. And it doesn't even need to be like data that they have publicly available, like in a data sheet. Like I can do things with, say, WebPlot Digitizer. You can actually extract data from plots, images, and maps, and get information about what they did from just a graph. So for example, this is what I was working on actually last month. I was able to take this graph here and get general information about an experiment. So it's really cool stuff you can just do even with graphs. So with all that said, let's wrap up with some considerations here. So obviously, your more involved experiments will have more information to consider. A lot of times use pretty simple scaled down examples. There might be more elaborate things like time being involved, like repeated measures, survival, time to event, etc. There might be fixed effects. These are effects that you're not actually interested in measuring, but you want to control for because you know they might have an effect on something, like age, sex, ethnicity, etc. Or there might be random effects, things that are more along the lines of like location, like plot or batch, things that sort of have inherent variation that you want to also control for. Another consideration, there might be missing data, and you might have to figure out methods to work with that. And of course, anything else that would quote unquote make the analysis harder. It's good to know those ahead of time. And really, with all this, thinking through all these topics, it's really better for the researcher to be able to start to answer these questions themselves, have an understanding of that, than the statisticians to try to answer them by assuming certain things or sort of distilling out what they think the researcher is trying to do, rather than the researcher telling them as clear as they can what they want to do. Because the researcher, of course, has the most intimate knowledge of the research. That, they have the most vested interest. They played around with the most. They're in the best position to be able to answer such questions. And of course, with anything, you don't have to have all the answers. A lot of times, you might not figure things out until you actually do it, put things into action. But it's always useful to start with a basic setup, start with a basic plan, and then add complexity from there. And, any other sort of considerations, there's a whole kitchen sink worth of analyses that can cover such things. All right, what's next here? So with my closing points, these 4 big topics are meant to help a statistician understand the research project, help a researcher understand, frame, and communicate the research to that statistician, so that there's clear, helpful, and useful communication between the two. Because really, the clearer the design of an experiment, the better the chance for a successful outcome. And a well-defined data plan, a plan for like what the data is going to be, how it's going to be analyzed, it really protects against failure due to faulty planning. Like, for example, maybe you need a variable in your analysis, but if you forget to include to measure it, you might do a whole experiment and you can't go back and measure something when it's already done. Or maybe you don't have enough samples, or maybe, so on and so forth. So it helps prevent against failure. And really, hopefully this also helps researchers just on their own be able to think about their data and help better plan their experience, even outside consultation with the statistician. So yeah, I really hope that this talk has been able to help bridge the gap between the statistician and the researcher. And now what I have for you outside of this talk here is if you found this useful, it'd be really helpful if you would take a survey about this talk here to see how we did. And then also, I've actually designed a questionnaire based on these 4 big topics, so that if you are a researcher yourself and you want to use this either in consultation with me or other statisticians, use this as you see helpful. And as a statistician for the DaCCoTA project, I obviously can help with research. And just a quick rundown here of who and what I can support. Obviously, if you're part of the DaCCoTA CTR, either career development or pilot projects, obviously take precedence. Though if you're part of any of the DaCCoTA institutions like UND, NASU, etc., you do federally funded research, general consultation. I've worked on various people in and out of my network, grant application, industry funded, all the way up down to even like unfunded research, as long as there's time. So the BERD Core is available to kind of walk through these questions with you. With that, thank you for your time. Again, we're the DaCCoTA project, supported by a NIH grant, so please cite us if you found this useful in any way. All right, have a great day.